Hi, Shane Monty from the Buzsáki lab here. Today I'm going to show you how to use silicon probes to perform high-density electrophysiological recordings of unit activity and local field potential in the awake behaving rat. I'll show you how to make a small microdrive, affix a silicon probe to it, and then implant the probe in the brain for chronic recordings. This process is pretty straightforward, but does involve a number of steps, so let's get started.
Here is a small collection of microdrives for rats and mice for moving multiple single wires, tetrodes and silicon probes that we have designed and fabricated over the years. Silicon probes can be attached to rigid printed circuit boards or flexible polyimide cables. We will show you here how to affix this 4-shank probe to a microdrive. To prepare a drive, we need a brass plate, drill bit size 55 and 65, a tap, a single row pin header, a 0090 screw and corresponding nut, a drill bit holder, and a screwdriver. The first step is to break a 3-pin piece from the header. Gently pull out the middle pin. Enlarge the hole by drilling through it with a drill bit size 65.